Hello students, welcome to this session where we are going to discuss autoimmune diseases and uveitis. To understand autoimmune diseases and autoimmunity, we need to understand how the immune system works under normal conditions. You can compare your immune system to the defense forces defending your country. So just like the candidates are recruited, they're trained and then they're given specialized training and deployed to different parts of the country where they're either involved in active patrolling or they're held in reserve to respond to a threat. Just like that, the cells of the immune system are also trained, they're given specialized role and they're sent to different parts of the body where they identify a threat, they decide which cells are best to neutralize the threat, neutralize it, and then repair and restore the body to its normal condition. So in this session, we are going to look at the different cells of the immune system, their structure and function, and the different types of immunity we have, namely innate and adaptive immunity. Now just think about it. Your body is actually surrounded by a sea of microbes. The microbes are there in the air we breathe, it is there in the water we drink, and the food we intake. Yet, for most parts, we are healthy. And this is because of a very alert immune defense system. So there are three tiers of defense in our body. The first is the physical barrier. So wherever your body is exposed to the external environment, say like the skin, the digestive tract, the respiratory tract, you're lined with epithelia. And these provide a very important mechanical barrier to invasion of microbes. When they break down, you're at a higher risk of getting infection. To give you an example, if there is a corneal epithelial defect, you're certainly worried about getting a secondary infection. So it's not just that they provide mechanical barriers, they also have other mechanisms like the cilia in the respiratory tract. So what they do is they actually sweep out the dust and the foreign particles. Or take the example of the eye, you have the tears and the blinking phenomenon, again which helps to clear out dust and foreign particles. In addition to this, they also have chemicals which provide protection against invading microbes. A classical example would be the lysozyme, which is present in abundance in tears and saliva. So suppose the microbe manages to breach the physical barrier, the innate immunity kicks in. And what you mean by innate immunity is it's immunity that you're born with, so it's not learned. And it has both cellular and molecular components. So the two characteristics of the innate immune response is that it is very quick, very rapid, but it is not specific and it doesn't have memory. If the innate immunity is not able to control the infection, or if it is severe enough, the adaptive immunity comes into play. And this is mediated by the T and B lymphocytes. Okay? So in contrast to the innate immunity, the response here is more specific and it also provides memory. By that what you mean is, if the pathogen infects you a second time, the immune system remembers it and comes up with a faster and more vigorous response and clears the infection faster. Now let's look at the cells of the immune system. They arise from a hematopoietic stem cell in the bone marrow and they have two lineages, namely the lymphoid and the myeloid. The lymphoid lineage differentiates into the B cell, the T cell and the natural killer cell. The B cell can further differentiate into the memory B cell and the plasma cell which produces antibodies. The T cells has several subsets like the helper T cell, the cytotoxic T cell, the memory T cell and the T regulatory cell. If you look at the myeloid progenitor, it differentiates into the granulocytes, namely neutrophil, eosinophil and the basophil. The tissue counterpart of basophil is called the mast cells. This also includes the monocyte, macrophage and dendritic cell which constitute the mononuclear phagocyte system. Now let's first look at the granulocytes. Okay? This consists of three cells, namely neutrophil, basophil and the eosinophil. They are called granulocytes because the cytoplasm contains abundant granules. They contain several different enzymes and depending on the color of staining of these granules, they are classified into neutrophil, basophil or eosinophil. They are the most abundant cells in the immune system. They constitute almost 2/3 of the WBCs and they are largely responsible for the innate immunity. They are also the first responders in case of inflammation. Now let's look at the neutrophil in some detail. This is the most abundant WBC you see. It's also the smallest. Its nucleus generally has multiple lobes, anything from 2 to 5. And you can see the granules here in the cytoplasm. They stay neutral pink in the hematoxylin eosin stain, hence the name neutrophil. The neutrophils are short-lived and they generally move around in the circulation. But whenever there is a threat or an invasion or an infection, they move to the site of the infection by a process called the chemotaxis. And these are usually the first responders in inflammation and they're very important in fighting bacterial infection. So there are three mechanisms by which a neutrophil can destroy an invading microbe. The first and foremost is phagocytosis, where the bacteria is actually engulfed by the neutrophil. The cell membrane pinches off, forming what is called a phagosome. This then combines with what is called the lysosome, which is an organelle present in the cell and is actually the digestive tract of a cell. The lysosome contains several enzymes which can break down protein, nucleic acid and lipids. So after it combines, the enzymes are discharged and they destroy the bacteria. The second mechanism is by degranulation of the granules in the cytoplasm, which can kill the bacteria. And the third and a very interesting way in which it can attack invading bacteria is by what is called netosis or neutrophil extracellular traps. Here what happens is the nuclear chromatin is discharged out in the form of a net. And this net also contains antimicrobial peptides and enzymes. They actually limit or kill bacteria as well as fungi. So the next granulocyte we need to look at is the eosinophil. So unlike the neutrophil, the nucleus here is bilobed. Okay? And the cytoplasmic granules here stain red with eosin. Like neutrophils, these cells can also destroy microorganisms by phagocytosis. Now, when these cells are stimulated, the cytoplasmic granules are discharged, releasing a number of different molecules like chemokines, cytokines, lipid mediators, and growth factors. Now, historically, it is known that these cells are important in hypersensitivity or allergic reaction, and also important in defending against parasitic or helminthic infection. But now, in addition to these roles, Eosinophils also have been found to have a role in immunomodulation, tissue repair, and remodeling. Coming to the last of the granulocyte, namely the basophil, is the largest of them all and the least common. And it has a bilobe nucleus. It's called basophil because the granules in the cytoplasm stain blue. So you can remember basophil for blue. So the basophils, like the eosinophils, are important in defense against ailments and ticks. And it's also important in hypersensitivity reaction. In addition to that, these cells secrete Ig4, which helps in transforming naive T cells to T helper 2 cells. Moving on to the mononuclear phagocyte system, it consists of 3 different cells, namely monocytes, macrophages, and dendritic cells. They play a very important role in the innate immune system. Now, traditionally, it was thought that the monocytes in the circulation moves into the tissue and differentiates into macrophages and dendritic cells. But now it has been found that they might have a separate embryonic origin in the case of macrophages, and the dendritic cells might have a separate origin in the hematopoietic stem cells. The important function of monocyte include phagocytosis, antigen processing and presentation, and releasing cytokines. When the monocyte transforms into macrophage, it's almost like an ordinary person transforming into a Spider-Man or a Superman. The cell enlarges 5 to 10 times its normal size. The intracellular granules increase in number as well as its complexity. You have much higher levels of cytokines and enzymes, and the ability to phagocytose is increased. The macrophage is lodged in different parts of the body, and depending on where it is, it's named differently. So in the brain, you have the microglial cell. In the liver, you have the Kupffer cell. In the kidney, you have the mesangial cell. In the skin, Langerhans cells. And the bone, it is called the osteoclast. So the macrophage is a very versatile cell, with multiple roles to play. It's important in both innate and adaptive immunity. So in innate immune reactions, it's important in destroying microbes by phagocytosis. You remember the neutrophils which we explained earlier. It also removes the dead cells, and it regulates immune response through interleukin 10. In adaptive immunity, it's very important in signaling and amplifying the immune response. As one of its most important function is to do antigen presenting to T cells, and thereby it activates both T and B lymphocytes. So let's look at how macrophages act like antigen-presenting cells. So you have the antigen, which is taken up inside the macrophage. It's broken down, and a piece of the antigen is attached to MHC class 1 protein, and this then moves to the cell surface and it's displayed. This combination of MHC and the antigen is then recognized by the appropriate T cell, resulting in its activation. Now, macrophage also has several functions to perform in health. So it's responsible for a lot of tissue repair and remodeling, that it's in bone, in the hematopoietic system, in the synapses. It's also involved in wound healing and tissue regeneration. Now, moving on to the lymphoid line of cells. These cells are found in both the blood and the lymphoid organs, and they arise from a stem cell in the bone marrow. They play a key role in the adaptive immune response. There are mainly 3 types. You have the T lymphocyte, the B lymphocyte, and the natural killer cells. Of these, the natural killer cell is mainly involved in the innate immune response. So let's look at T lymphocytes. They originate in the bone marrow, but they move to the thymus for maturation, and hence the T of the T lymphocytes. They have a receptor called the T cell receptor, which is very specific for an antigen, and it can recognize only a MHC-bound antigen. It will not recognize a free antigen. And the T lymphocyte is recognized by the presence of CD3 glycoprotein on the surface. So the subsets of T lymphocyte which we need to be familiar with are the helper T cell, and the cytotoxic T cell, and the regulatory T cell, which plays a huge role in adaptive immunity. Now, one of the terms you're going to come across frequently in this context is CD4 positive and CD8 positive. What CD means is cluster of differentiation, and these are a group of cell surface proteins which are useful for identifying the lineage and the maturation status of a lymphocyte. So the helper T cell is CD4 positive, and it will recognize antigens which are presented along with MHC class 2. Whereas the cytotoxic T cell is CD8 positive and will recognize antigens which are presented to it along with MHC class 1. So the T cell is central in adaptive immune response. Once it's activated, it sets off a series of reactions resulting in activation of B cell and antibody production. It recruits and activates the mononuclear phagocytic cells. It also helps in activation of cytotoxic T cells, and it secretes a lot of cytokines which are responsible for further growth and differentiation of T cells, monocytes, and macrophages. And once the inflammation is controlled or the invading microbe is cleared, it helps to restore the immune status to normal. So it regulates the immune reaction. Because of this reason, it's very important that the T lymphocytes should not be activated randomly. So there are several checkpoints or several steps to activation of a T lymphocytes. For one, the T lymphocyte cannot be activated by a free antigen. It has to be presented by a antigen presenting cell. So here you have a dendritic cell presenting the antigen along with the appropriate MHC to a naive T cell. It then proliferates, undergoes clonal expansion, and then differentiates into helper T cell, cytotoxic T cell, or the memory T cells. Now the B lymphocyte, unlike the T lymphocyte, it originates from the bone marrow and undergoes maturation in the bone marrow itself. So the B stands for bone marrow, although originally it actually stood for the bursa of Fabricius. This is MHC class 2 restricted, and it has got a receptor on the surface called the B cell receptor, much like the T cell receptor. So the B lymphocyte is responsible for the humoral immunity by production of antibodies, and these antibodies, they can target specific antigens The B lymphocytes can also act as an antigen-presenting cell to T lymphocytes, resulting in T cell activation, which in turn can activate more B cells and help them differentiate to plasma cells. So there are two mechanisms by which a B lymphocyte can be activated. One is the T cell independent mechanism. Okay, here you have a large molecule with multiple epitopes, and this can cause a cross-linking of the receptors on the B cells. They get activated, get transformed into plasma cells, which produce antibodies. But the important thing here is there is no memory here. On the other hand, if you have a protein antigen, which doesn't have multiple epitopes, you need a T cell dependent activation of the B cell. So the B cell will act like an antigen presenting cell to the T lymphocytes, which gets activated and in turn activates the B cell, which undergoes clonal expansion, and you get plasma cells producing antibodies. Here, unlike the T cell independent one, you get memory cells as well. Finally, we come to the natural killer cells, which is the third subset of lymphocyte, and this one, unlike the other two, is important in innate immunity. Now, what makes it unique is that it does not have a specific antigen receptor. It doesn't require any antigen to be presented to it. It's capable of recognizing an abnormal cell by the reduction in the MHC class 1 on the surface or an unusual surface antigen. And it can directly kill cells which are infected with viruses or which have tumor. So here is a diagram which shows the difference between the killing mechanism of the natural killer cell and the T helper cell. So in the T helper cell, you have a microbe which is being ingested by the antigen presenting cell. It's presented to the T cell along with the appropriate MHC class, which then gets activated. But unlike that, the natural cell doesn't require an antigen presentation. It recognizes when a cell is abnormal and directly destroys it. So it's very important in providing defense against viruses and tumors through antibody-dependent cell-mediated toxicity. It also secretes cytokines. Now let's look at some of the important difference between the T and the B lymphocytes. We know that the B lymphocytes matures in the bone marrow and the T lymphocytes in the thymus. B lymphocytes are mainly responsible for the humoral immunity, whereas the cell-mediated immunity is mainly due to the T lymphocytes. The B lymphocytes have subsets called memory cells and the plasma cells, whereas T lymphocytes can differentiate into the helper T cell, the cytotoxic T cell, the memory cells, and the T regulatory cells. The B lymphocytes produce antibodies and the T lymphocytes produce cytokines. The cell surface marker for B cell is the CD19 and that for T cell is CD3. And the receptors are different. You have the T cell, you have the T cell receptor, and the B cell receptor is actually an immunoglobulin. So when it comes to its function, the B lymphocyte does not require antigen processing to get activated, whereas it's always required in the case of T lymphocytes. The B lymphocytes binds with extracellular antigens, whereas T lymphocyte will bind with foreign antigen which has been combined with the MHC molecule. The B lymphocyte eliminates free foreign invaders, whereas the T lymphocytes will lyse virus-infected cells and tumor cells. So before we finish, let's look at the different components of the immune system. So you have an innate immune system and an adaptive immune system, each of which has cellular components and molecular components. So in the innate immune system, you have the granulocytes, namely neutrophil, eosinophil, basophil, and the mast cells, and you have the macrophage, monocyte, and the natural killer cells. The molecular component of the innate immune system includes mainly complements, cytokines, and chemokines. The T and B cells form the cellular component of the adaptive immune system, and the antibodies are responsible for the molecular component. So in this session, we have seen the different cells of the immune system and how they get activated and its role in normal immune responses. I hope this session has been useful. Thank you.